When you think about bacteria, what's the first thing that pops into your head? Maybe germs, something you want to steer clear of.
Yeah, that's probably pretty common.
But the truth is, bacteria are absolutely everywhere. I mean, they're living deep in the Earth's crust. They're in every drop of water floating in the air. And yes, they're absolutely all over and inside other living things, including us.
They're fundamental.
And their impact on the planet and on us is enormous, right? And it's not just about causing illness. While some bacteria definitely cause problems, so many others are incredibly important, vital, even for our health, our environment, and even the food we eat.
That's such an important distinction to make. There's this complex, bustling microbial world that surrounds us and is part of us, and we're really only beginning to understand the full scope of their interactions.
Right.
And increasingly, the focus isn't solely on getting rid of potentially harmful bacteria, but on understanding and promoting the beneficial ones. There's this fascinating shift towards seeing how we can work with these tiny organisms.
Like allies instead of enemies.
Exactly. Even finding ways to introduce specific beneficial strains to potentially replace harmful ones or support our health.
It's a total game changer in how we think about these microorganisms, isn't it? Instead of just thinking of them as invaders, we're exploring them as allies. And some of the science is, well, it's mind-blowing, like research into bacteria genetically engineered to perform a specific health task, and then once it's done, essentially disappear.
The level of control is increasing.
These incredibly small organisms have effects that are anything but small. They influence everything from agriculture to human health, and there's still so much to uncover.
Absolutely. And to truly understand them, especially the beneficial ones we often hear about now, the probiotics, we face a fundamental challenge. We have to be able to see them.
Which sounds simple, but
Given their microscopic size, this is far from simple. It's a real technical hurdle.
which is exactly where we're diving in today. This is a deep dive into the fascinating world of bacteria, zeroing in on those helpful probiotic strains.
Yeah, we're going deep on this one.
We're going to explore the cutting-edge techniques scientists use to visualize these minuscule life forms, follow the incredibly difficult journey a probiotic takes once you consume it, look at how these bacteria attempt to stick around inside your body.
And even peek inside the genetic blueprint of a specific probiotic strain to understand what makes it tick, see what it's capable of, essentially.
Exactly. And everything we're talking about today, all the insights and details are drawn directly from the sources provided for this deep dive.
Right, we're sticking to the research, distilling those key takeaways.
So let's start at the very beginning. How do you even begin to visualize something that's invisible to the naked eye? What tools do scientists use?
Well, that's precisely the first hurdle, their size. Bacteria are measured in micrometers, sometimes even nanometers.
Yeah.
Tiny.
Right.
You can see them with a basic light microscope, sure, and our sources actually show some images of bacteria in foods like yogurt taken that way. But if you want to understand their detailed surface structure, their shape, how they look really up close,
You need something more powerful.
you need something much, much more powerful.
And that's where techniques like scanning electron microscopy or SEM come in, right?
Precisely. SEM is an incredibly powerful tool. It allows scientists to capture these really detailed images of the surface of a specimen.
So not looking through it, but at its surface.
Exactly. Think of it like taking a super high-resolution 3D photograph of something extremely small. It reveals the intricate topography of the bacterial cell surface, how it's shaped, its texture, how it might physically interact with its surroundings. It's all about the external details.
That sounds amazing. So you just pop a sample under this super microscope, and there they are, ready to view.
Oh, if only. Getting a sample ready for SEM is a complex, really painstaking process. It requires multiple steps, and often these steps have to be tailored specifically to the type of material you're looking at.
Okay.
The sources go into quite a bit of detail about this, actually. It shows that the preparation is really half the battle.
What kind of samples are we talking about? Can you really look at bacteria in anything? Or are there limits?
Well, the techniques described cover a pretty wide range of specimens. You can prepare samples from simple things like soil or water, or from various food products like yogurt, cheese, or even meat like ham.
Okay.
And they also detail methods for looking at bacteria inside living organisms, so samples from, say, chicken intestines or even human feces. But each material requires careful initial handling. You can't just treat everything the same.
Okay, so how do you start with something like a piece of cheese or some yogurt? They seem pretty different.
They are. For solid things like cheese, you need to trim them down into very small pieces. Tiny little prisms, actually. The source mentions dimensions like 10, 15 millimeters long, but less than 1.5 millimeters square in cross-section. Really small.
Why so small?
So the preparation chemicals, the fixatives and dehydrating agents, can actually penetrate properly and quickly. For more liquid or viscous samples, like yogurt or cream, you might need to encapsulate them first in a gel, like an agar gel.
So you trap them in a gel.
Yeah, it gives some structure, makes them solid enough so they can be handled through the rest of the process without falling apart. And for things that might be damaged by heat, like, say, raw egg yolk, they use a different type of gel, calcium alginate, which works at lower temperatures.
And if you've grown bacteria in the lab on a dish.
If you've got colonies on an agar plate, you carefully cut out a small piece of the agar with the colony sitting on top, maybe leaving a little clear rim of agar around it. The key, really, is getting a tiny, stable piece of your specimen to work with.
Once you have that tiny piece, what's the main goal of the first chemical steps? What are you trying to achieve?
The absolutely critical first step is called fixation. The goal here is to instantly preserve the structure of the bacteria and their immediate environment exactly as they were, like freezing a moment in time.
Ah, okay.
This prevents them from degrading or changing shape during all the subsequent processing steps, the washing, the drying. They use chemicals, typically glutaraldehyde, that cross-link proteins and lock the cellular structures in place.
And I imagine different things in the sample might need different chemicals to fix properly, not just proteins.
That's a really important point the sources highlight, yes. While glutaraldehyde works well for proteins, if your sample contains significant amounts of fats, like fat globules in milk or cheese or certain complex sugars, like those found in the mucus lining our gut.
Which is super important for probiotics.
Exactly. You need extra steps or different chemicals to make sure those components are also fixed and retained. Otherwise, they might just wash away or collapse during prep.
So what do they use for fats, for instance?
For lipids, they often use osmium tetroxide, sometimes combined with other chemicals like imidazole. This reacts with the fat molecules and helps them stay in place, preserving those fat globules you see in cheese or yogurt.
And for mucus, you mentioned that washing away could be a problem.
Right. If you want to see bacteria actually attached to the mucus lining the intestine, which is, as you said, super relevant for probiotics trying to stick around, conventional methods might just wash the mucus and the attached bacteria right off.
Yeah, you'd miss the whole interaction.
Completely. So they add special dyes or chemicals like ruthenium red or Elchian blue directly to the glutaraldehyde fixative. These substances help stabilize and fix the mucus structure itself, keeping it, and hopefully the bacteria stuck to it, in place.
That sounds like a crucial detail, especially for studying how probiotics interact with our gut lining. It really shows the level of specific detail required just to get a reliable image.
It absolutely does. And getting these chemicals to penetrate all the way through denser samples, like a piece of cheese or ham, can take a long time. The sources mention needing hours, sometimes even a full day, for proper fixation. They even describe a test where they cut the sample after different fixation times to see how far the dark fix zone has penetrated. Yogurt fixes faster than cheese, apparently.
Okay, so fixed, preserved in time. What's next in the pipeline?
After fixation, you carefully wash away the excess fixing chemicals. Then you have to remove all the water from the sample. This is called dehydration.
How do you get the water out without everything collapsing?
You do it gradually. The sample is soaked in a series of solutions with increasing concentrations of alcohol, usually ethanol. So you go from, say, 50% ethanol to 70%, 90%, 95%, and finally 100% or absolute ethanol. It's like slowly replacing the water inside the sample structures with alcohol, molecule by molecule.
Okay, gradual replacement, and once all the water is gone, replaced by alcohol.
Then comes another absolute rule mentioned in the sources. For conventional SAM, the specimen must be completely dry. Any water left that evaporates during observation in the microscope's vacuum would create strong surface tension forces that could shrink or collapse the delicate structures you want to see.
So how do you dry it safely? You can't just let it air dry, right?
No, exactly. Air drying would cause too much damage. They use a special technique, most commonly critical point drying, or CPD. This involves replacing the alcohol with liquid carbon dioxide under pressure, and then carefully heating it above its critical point where the liquid and gas phases merge. Then you can release the CO2 as a gas without it ever passing through a liquid-gas boundary, which avoids that damaging surface tension.
Wow, that sounds complex.
It is a bit technical, but it's the standard way to get beautifully preserved, dry samples for CM.
Okay, so you've preserved it, dehydrated it, and dried it carefully. But what if the bacteria you're interested in are inside something solid, like buried within that cheese matrix you mentioned? How do you see them?
That's where you need to fracture the sample, essentially break it open to reveal the internal structure.
Snap it.
Kinda, but there are a couple of main ways to do this, and they give different results. You can do what's called dry fracturing. You take the sample after it's completely dried, and then you physically break it. This tends to fracture along the naturally weakest points in the structure, so you get a rough, uneven surface. The advantage is that the bacteria themselves usually remain intact on this surface.
And the other way.
The other method is freeze fracturing. This is usually done after dehydration in absolute ethanol, but before the final critical point drying step. You rapidly freeze the ethanol-infiltrated sample, often in liquid nitrogen, and then fracture it while it's frozen solid.
How does that look different?
Freeze fracturing often produces a much smoother, flatter fracture surface. This can be really good for evaluating the overall internal structure. You can see the distribution of things like bacteria, yeast cells, starch granules, fat globules, maybe even void spaces within the matrix. The downside is that any bacteria caught right on the fracture plane will also be broken open. So you get a different kind of view, more like a cross-section. The source mentions this technique was used for an image of lactic acid bacteria inside cottage cheese.
So different fracturing methods give you different perspectives on the internal structure and the bacteria within it. Fascinating. Once you've got your tiny fixed, dried, maybe fractured sample, how do you actually put it under the microscope?
You need to mount it securely onto a small metal holder called an S-stub, usually made of aluminum. You typically use double-sided sticky tape on the stub face to position the fragments. Tape to hold it, but then, importantly, you need to make it electrically conductive. The SIM works by scanning a beam of electrons over the surface, and you need the electrons that hit the sample to be able to flow away to the ground. If the sample isn't conductive, electrons build up, it gets charged, and that really messes up the image.
So how do you make, say, a piece of cheese conductive?
After sticking it down, you usually paint a conductive material, like silver paint or carbon cement, onto the sides of the sample fragments, making sure it creates a continuous path from the top surface down the sides to the metal stub itself. The source gives a little practical tip here. Too thick paint won't adhere well, but too thin and it might soak into porous samples and cause artifacts.
Artifacts, like things that appear in the image but aren't actually real biological structures. You mentioned those might happen.
Exactly. And the sources mentioned some really interesting examples, especially in food microscopy, where artifacts initially led to misinterpretations. There were these apparent filaments seen in some yogurt SEM images.
Filaments, like threads.
Yeah, thin threads connecting bacteria to the protein matrix. Scientists initially thought maybe they were structures the bacteria used to attach or some kind of interaction, but they weren't. Turns out they were likely artifacts. They think the polysaccharide capsules that surround some yogurt bacteria weren't properly fixed by the standard methods. During the dehydration and drying process, these unfixed capsules probably shrank and collapsed into these thin, thread-like structures. So not a real bacterial appendage, but a result of the prep.
Wow. And you mentioned void spaces, too.
Yes, another example. Sometimes you see these empty gaps or voids around chains of bacteria in the yogurt protein matrix. Again, initial speculation was maybe the bacteria were actively keeping the casein protein particles away as they grew.
Plausible, maybe?
Plausible. But the more likely explanation, based on the source, is again related to those unfixed polysaccharide capsules. While the yogurt was setting, the capsules occupied space, pushing the protein particles apart. When the capsules were then removed or collapsed during SEM preparation, they left behind these empty spaces, these voids. So an absence created by the prep, not an active process by the bacteria.
Wow. So seeing these microscopic images isn't just about looking, it's about sophisticated interpretation. You have to constantly ask, is this real or is this an artifact of how we prepared the sample?
It's a crucial point. Understanding the potential artifacts is just as important as mastering the preparation techniques themselves. You need to be critical of what you see.
It really highlights the expertise involved. Are there special tricks for getting bacteria out of liquids, like water samples or suspensions?
Yes, a common way is to capture them on filters. You might do some preliminary filtering to get rid of larger debris first. Then you vacuum the liquid through a fine-pored filter membrane, usually a small circular one, maybe 13mm diameter, that fits nicely onto an F-stub. The bacteria get trapped on the filter surface.
Can you fix them before or after filtering?
You could do either. Capture them live and then fix them on the filter, or fix them in the liquid first and then filter. The source actually shows a setup using a tilted filter holder and a pasture pipette to carefully load the bacterial suspension onto the filter, trying to get an even layer.
And they mention a practical warning about mounting these filters.
They do. A really useful one. If you use double-sided sticky tape to cover the whole back of the filter to stick it to the stub, and you plan to store it for days or longer,
Yeah.
the adhesive from the tape can slowly penetrate through the pores of the filter and eventually affect the bacteria on the top surface, potentially creating artifacts or damaging them.
Oh, that could ruin your sample after all that work.
Exactly. So the recommendation is to just use small squares of tape at the very edges of the filter and then use conductive silver cement to properly secure it and provide conductivity. Filters mounted that way can apparently last for weeks. Little details like that make a big difference.
Definitely sounds like it. They also mentioned something about magnetic beads.
Ah, yes, that's a clever technique. Newer, perhaps. It's a way to isolate specific types of bacteria from a complex mixture, like trying to find salmonella in a food sample.
How does that work?
You use tiny magnetic beads, usually made of polystyrene, that are coated with antibodies. These antibodies are chosen because they specifically bind only to the surface antigens of the target bacteria you're looking for.
So the target bacteria stick to the beads?
Precisely. You mix the antibody-coated magnetic beads into your sample liquid. The target bacteria bind to the beads. Then, you just use a strong magnet held against the side of the tube to pull the beads with the attached bacteria out of the suspension, leaving everything else behind.
That's neat, like fishing for specific bacteria.
It is. Then you can take that small, concentrated volume containing the beads and captured bacteria, maybe filter it onto a small nuclear pore filter, and prepare it for SEM, just like any other suspension. It lets you focus your view on just the bacteria you're interested in.
That makes sense. And what about biofilms? Bacteria sticking together on surfaces.
The sources briefly mentioned biofilms, noting that they're a growing area of interest. These are communities of bacteria attached to a surface and encased in a slimy matrix they produce. The challenge for SEM is that these structures can be quite delicate and easily disturbed during sample collection and preparation, so getting a true representation can be tricky.
Okay, so SEM gives us this fantastic surface view, but you mentioned earlier, what about seeing inside the bacteria? Is another microscopy method mentioned for that?
Yes, transmission electron microscopy, or TEM, is briefly mentioned, unlike SEM, which bounces electrons off the surface to create an image.
CAM goes through.
Right. TEM shoots a beam of electrons through an extremely thin slice of the sample. Where the electrons pass through easily, the image is bright. Where they're scattered or absorbed by dense structures inside the cell, the image is dark. This allows you to see the internal structures, things like the cell wall, membrane, ribosomes, DNA region.
So it gives a different perspective.
A completely different perspective. While the main focus of the SEM prep discussion was surface detail, TEM is another incredibly powerful tool for studying bacteria. It provides information about their internal organization and morphology. For example, the TEM image described for the TY11 strain later on was used to measure its cell length and width, giving basic morphological data.
Got it. Now, beyond the classic black and white sort of flat images we often associate with electron microscopy, can these images be presented in ways that show more information or maybe just look more visually appealing?
Definitely. The sources discuss a couple of interesting techniques for enhancing the visualization. One is creating stereo images.
Stereo, like 3D.
Exactly. You take two separate SEM images of the exact same area, but you tilt the sample slightly. The source suggests between 6 and 12 degrees between taking the two shots.
And how does that make it 3D?
When you view these two images together, this stereo pair, either using a special stereoscope viewer or by combining them digitally into an anaglyph.
Ah, the red and cyan images you need the special glasses for.
Yes, those. Because each image was taken from a slightly different angle, just like your two eyes see things slightly differently, your brain confused the two images to perceive depth. It creates a strong three-dimensional impression of the sample surface.
That must be amazing for understanding complex structures.
It really is. You can really see the depth, how things are arranged spatially, how bacteria might be nestled in crevices or forming layers. It adds a whole new dimension, literally, to the visualization. The source even mentions needing to rotate the images because of how the microscope stage tilts, and that there's free software available to help create these anaglyphs.
That's cool. What about color? We often see these incredibly vivid color images of bacteria or viruses in magazines or online, but you said scientific papers are usually grayscale.
Right, that's called false coloring. In scientific publications, SEM images are typically presented in grayscale. That grayscale directly reflects the intensity of the electrons detected from each point on the sample. It's the raw data, essentially.
So the colors aren't real.
No, bacteria don't have vibrant colors like that at the microscopic level. The color you see in popular science articles, educational materials, or documentaries is added afterwards. It's false color or pseudo-color.
Why do they do that? Is it just to make them look pretty, catch the eye?
Partially, yes.
Yeah.
The visual appeal is definitely a factor. Bright, colorful images are more attractive to art directors and readers than plain grayscale. But the sources emphasize that false coloring is also a valuable tool for communication. It can make complex or difficult subjects much easier to understand, especially for a non-expert audience. By assigning different distinct colors to different types of bacteria in a mixed population, or to different structures within a sample, or maybe highlighting bacteria versus the background matrix.
Ah, it helps you distinguish things visually.
Exactly. It clarifies what you're looking at, guiding the viewer's eye and helping them interpret the image more easily. For example, coloring different bacterial species, different colors in that feces sample image, makes it instantly clearer that there's a mixed community present.
How is the coloring actually done? Is it automated or is it an artistic process?
It's generally a skilled manual process.
Mhm.
It's done using graphic software, like Adobe Photoshop as mentioned. The artist, often a specialized microscopist or scientific illustrator, will carefully select different areas or structures in the original grayscale image, maybe based on shape, size, or texture, and then assign specific colors to those selections.
So they're making interpretive choices.
They are. For complex images, like that fecal sample with multiple bacterial types, the source mentions they might work with layers in the software. So they might select all the bacteria that look like type A and put them on one layer, color it blue, select all the type B bacteria onto another layer, color it green, and so on. It allows for precise control.
So while the colors aren't the bacteria's natural appearance, the coloring itself isn't random. It's done deliberately to highlight biologically relevant distinctions and aid interpretation.
That's the idea. It's a tool to enhance understanding. And the sources even point out that you can sometimes get these pre-colored images from specialized microscopists or from scientific photobanks.
It really is incredible how much effort and ingenuity goes into simply being able to visualize these tiny organisms and then present that information clearly and effectively. It's absolutely foundational to understanding them, isn't it?
It absolutely is. You simply can't study what you can't see properly. All the downstream analysis depends on getting good, reliable images and interpreting them correctly.
Right. So we've seen how scientists can get a good look at bacteria using these sophisticated techniques. But let's shift focus now to those beneficial bacteria, the probiotics. What happens to them after we consume them? They're introduced into a completely different and often quite hostile environment inside us.
That's the crucial next step in their story. Probiotics, as defined by the FAO and WHO back in 2001, are living microorganisms which, when administered in adequate amounts, confer a health benefit on the host.
Okay, and that definition itself holds the key challenges, right? Living and adequate amounts.
Exactly. They need to be living when they reach the part of the body where they exert their effect, which is typically considered the colon, the large intestine. And they need to arrive there in adequate amounts, enough cells to actually make a difference.
So they basically have to survive a kind of obstacle course through the entire digestive tract.
That's a great way to put it. The sources describe this as a long journey, and it presents really significant challenges that can drastically reduce the number of viable, living probiotic cells that actually make it to their destination. Many just don't survive the trip.
What are the main obstacles they face, starting right from the moment you swallow them?
Well, first they pass through the mouth, where saliva has some antimicrobial properties, but the main action starts soon after. The first major hurdle is the stomach.
The acid bath.
Precisely. The stomach is designed to break down food and kill ingested pathogens, and its primary weapon is highly acidic gastric juice. The pH in there is extremely low, maybe between 1.5 and 3.5. That level of acidity is lethal to many types of bacteria, including potentially many probiotic strains.
Okay, so that's a big filter right there. If some manage to survive the stomach acid and move into the small intestine, is it smoother sailing from there?
Not quite. Conditions do become less acidic once the food mixture passes the pylorus into the small intestine. The pH starts to rise towards neutral, maybe 6.8 or to 7.0, so the acid stress decreases. But the small intestine introduces a new set of chemical challenges. Bile acids, which are produced by the liver and released from the gallbladder to help digest fats, are dumped into the small intestine. Bile acids act like detergents and can disrupt bacterial cell membranes.
Okay, so bile is another threat.
Yes. And alongside bile, the small intestine is flooded with powerful digestive enzymes. Lipases to break down fats, proteases for proteins, amylases for carbohydrates. These enzymes aren't just breaking down food, they can also damage the bacterial cells themselves, attacking their membranes, their proteins, even potentially damaging their DNA.
So they're being attacked chemically from multiple angles.
They really are. The sources mention in vitro studies, lab tests simulating these conditions, using specific strains like Lactobacillus salivarius lyo-1 and Pediococcus panaceatus lyola-5. And these studies show a significant drop in the viability, the number of living cells, when these probiotics are exposed to simulated intestinal fluid containing bile and enzymes.
So intense acid in the stomach, then bile and digestive enzymes in the small intestine. It really sounds like a huge percentage of the probiotics you might swallow could be eliminated before they even get halfway through the system.
That's a very real possibility, yes. The loss can be substantial.
Now, if some hardy cells do manage to survive all of that and make it through to the colon, the large intestine, is that the end of the challenge? Are they home free then?
Not at all. Reaching the colon alive is a major achievement, but then they encounter perhaps the biggest challenge of all.
Which is?
The existing residents. The colon is the most densely populated microbial ecosystem on the planet. The numbers are staggering, something like 10,011 to 10,12 bacterial cells per milliliter of content. Orders of magnitude more concentrated than anywhere else in the body.
Wow. A crowded neighborhood.
Extremely crowded. And here, the primary challenge isn't so much the physical or chemical environment anymore, but biological competition. It's what scientists call colonization resistance.
Colonization resistance. That sounds exactly like the existing bacterial community defending its home turf, its established territory.
That's exactly what it is. It's a well-recognized phenomenon, actually discovered back in the 1950s. It describes how the stable, established community of your normal gut microbiota actively resists invasion by incoming bacteria, whether those are potential pathogens or, in this case, administered probiotics. They also resist the overgrowth of potentially harmful bacteria already present in small numbers.
So the locals fight back. How do they do this? What are their defense mechanisms against these newcomers?
The sources break it down into two main types: direct and indirect mechanisms. Direct mechanisms are things the resident bacteria do themselves to make life difficult for invaders.
Like what?
One key strategy is inhibition. Some gut bacteria produce their own antimicrobial compounds. A major class of these are called bacteriocins. These are protein-based molecules that specifically kill or inhibit the growth of closely related bacterial species, or species that are trying to compete for the same resources or ecological niche.
So like targeted chemical warfare.
Pretty much, yes. The source gives an example of bacteriocin-producing Enterococcus faecalis, inhibiting the growth of dangerous vancomycin-resistant Enterococci. The other main direct mechanism is simply competition.
Fighting over resources.
Exactly. The established bacteria are already highly adapted to that environment. They're efficiently consuming the available nutrients. They're occupying the prime real estate, the preferred adhesion sites on the gut wall or in the mucus. Any new bacteria arriving, like a probiotic, have to try and muscle their way in, find enough food, find a place to stick, all while competing against trillions of highly efficient residents. It's tough.
Okay, so direct inhibition and competition from the residents. What about the indirect mechanisms? You mentioned the host is involved.
Yes. Indirect mechanisms involve the host body interacting with or responding to the resident microbiota in ways that help maintain stability and resist newcomers. This can include the host producing its own antimicrobial peptides in the gut lining, maintaining the integrity of the epithelial barrier to prevent unwanted crossings, and even modulating the local environment, like influencing bile acid concentrations in ways that favor the established community. It's like the host and the home team are collaborating.
It sounds like a very robust defense system.
Oh.
So what does this intense colonization resistance mean for most probiotics that you take orally? Do they manage to set up shop?
Well, the general outcome, because of this strong resistance, is that most orally administered probiotic strains don't establish long-term residency. They tend to be transient. You can often detect them in stool samples shortly after someone starts taking them, showing they survived the transit to some extent. But they typically disappear quite rapidly from the feces. Once consumption stops, they're essentially flushed out. Some studies mentioned in the sources even show that despite regular probiotic intake, the overall structure and diversity of the person's underlying gut microbiota doesn't really change significantly.
That's a really important point, isn't it? It kind of contrasts with the popular idea that taking a probiotic instantly and permanently remakes your microbiome. It sounds more like they might have a temporary effect while they're passing through, perhaps.
That's often the current understanding, yes. A transient passage during which they might interact and have effects, but not necessarily long-term colonization for most strains in most people. And the sources also highlight another layer of complexity. Even this transient effect, or whether a probiotic can even be detected temporarily in the intestinal mucous membrane, seems to vary significantly between different individuals.
Really? Why would that be?
It's led to the concepts of permissive versus resistant individuals. Some people's gut environments seem more receptive, allowing certain probiotic strains to hang around a bit longer or reach detectable levels in the mucus, while others seem highly resistant, clearing them out very quickly.
What makes someone permissive or resistant?
It's likely a combination of factors: your individual genetics, the specific composition and stability of your unique baseline gut microbiota, maybe even your diet and lifestyle. It suggests that the success of a probiotic isn't just about the probiotic itself, but also very much about the host environment it lands in.
So it's not a one-size-fits-all situation.
Definitely not.
Okay, so even if a probiotic miraculously survives the acid, the bile, the enzymes, it still has to navigate this incredibly crowded, competitive, resistant environment in the colon. To have any sort of ongoing effect, they need to do more than just arrive. They need to somehow try and stick around, at least for a while.
Exactly. Establishing some level of persistence, which usually involves adhering to the intestinal lining, is considered absolutely critical. If they just pass straight through, their window for interacting with the host and potentially providing health benefits is very limited. So successful colonization, even if temporary, often hinges on adhesion.
What exactly are they trying to stick to inside our gut? What's the target?
They are primarily aiming to adhere to the intestinal mucosa. More specifically, they often target the thick layer of mucus that coats the epithelial lining of our intestines.
Mucus, like the slimy stuff.
Pretty much, yes.
Yeah.
The intestinal wall itself has layers: the epithelium, the lamina propria underneath, muscle layers. But the surface facing the inside of the gut, the lumen, is covered by this gel-like matrix called mucus. It's secreted by specialized cells in the epithelium called goblet cells.
And what's this mucus actually made of? Is it just slime?
Oh, it's much more complex than just slime. It's a sophisticated hydrogel composed mainly of water, but also contains large, complex, sugar-coated proteins called mucins. These are the main structural components. They link together, often via disulfide bonds, to form a tangled network that gives the mucus its viscous, gel-like properties. But it also contains slut-off cells, salts, lipids, proteins shed from cells, and even bacteria and bacterial products.
So it's a busy layer. Does it have structure?
It does, especially in the colon. The sources note that in the colon, the mucus often forms two distinct layers. There's a loose outer layer, which is less dense and is actually colonized by certain types of resident bacteria that are adapted to live there. Some even degrade the mucins for food.
Okay.
And then beneath that, closer to the epithelial cells, there's a tight inner layer. This one is much denser, more firmly attached, and generally considered to be largely sterile or only sparsely colonized, acting as a more robust physical barrier protecting the cells.
So the mucus is primarily a protective barrier, right?
Yeah.
Keeping things away from ourselves.
It definitely is a key protective barrier against physical abrasion from food, against large molecules, against direct contact with potentially harmful bacteria. But, and this is crucial for probiotics, it's also an ecological niche.
A niche. How so?
It provides a surface for bacteria to attach to, away from the main flow of gut contents. And the mucins themselves, those complex glycoproteins, can actually serve as a nutrient source for bacteria that have the right enzymes to break them down. So for bacteria adapted to it, the mucus layer is an excellent place to live. It offers protection, attachment sites, and food.
Okay, so it's both a barrier and a potential home. How do probiotics actually go about sticking to this mucus layer or maybe to the epithelial cells underneath? What's the process?
It's generally thought to happen in two stages. First, there's an initial reversible stage. This involves nonspecific physical forces, maybe just the bacteria bumping into the surface, or some weak general attractions based on things like surface charge or hydrophobicity, whether the surfaces repel or attract water. This initial contact is usually weak and easily broken.
So they might bump into it, but not really stick yet.
Exactly. The second stage is the crucial one for establishing stable attachment. This involves specific molecular interactions. Think of it like a lock and key mechanism. Specific molecules on the bacterial cell surface, called adhesins, recognize and bind to complementary receptor molecules on the host side.
And these host receptors could be on the mucus itself or on the intestinal cells.
Yes, both. Adhesins might bind to the mucin proteins in the mucus layer, or they might bind to specific sugar structures or proteins found on the surface of the intestinal epithelial cells, IECs, if they manage to get through or find gaps in the mucus. This specific binding creates a much stronger, more stable anchor.
And these bacterial adhesins, what kind of molecules are they? Are they all the same?
No, bacteria have evolved a diverse range of adhesins. They are very often proteins located on the bacterial cell surface. And this is where we find some really fascinating mechanisms that probiotics use. The sources discuss several types, and some are quite surprising. One interesting group is called moonlighting proteins.
Moonlighting proteins, like they have a day job and a night job.
Slight laugh. That's exactly the idea. These are proteins that are well known to have a primary function inside the bacterial cell. Maybe they're an enzyme involved in metabolism, like breaking down sugar, or perhaps they're a chaperone protein that helps other proteins fold correctly. That's their day job.
Okay.
But somehow, these proteins also end up on the outside surface of the bacterial cell. And once there, they perform a completely different, unrelated function, acting as an adhesin, helping the bacteria stick to host surfaces like mucus or epithelial cells. That's their moonlighting job.
Seriously, the cell just repurposes its internal staff for external work.
It seems so. The sources mention specific examples found in probiotics. Enzymes like NLase and GAPDH, glyceraldehyde 3-phosphate dehydrogenase, which are core metabolic enzymes, and also things like elongation factor 2, involved in making proteins, and molecular chaperones. All of these have been found on the surface of various probiotic strains, acting as adhesins, binding to mucins or host cells. Apparently, almost 400 such moonlighting proteins have been discovered across different organisms.
That's wild. Talk about multitasking. So besides these surprising moonlighters, what are some other, perhaps more dedicated sticking structures that probiotics might use?
Another key type of adhesin structure mentioned is pili, sometimes also called fimbriae. These are generally short, fine, hair-like protein filaments that extend outward from the bacterial cell surface.
Like little grappling hooks.
That's a good analogy. While pili are perhaps most famously known in pathogenic bacteria, where they're crucial for virulence and causing infections, they are definitely also found in many beneficial probiotic bacteria, including species of Lactobacillus and Bifidobacterium. They tend to be a bit narrower, maybe 110 nanometers wide, compared to some pathogenic pili, but they serve a similar adhesive function.
Are they all the same structure?
No, there's diversity here too. The sources go into some detail about the pili found on Lactobacillus rhamnosus GG, LGG, which is a very widely studied and commonly used probiotic strain. LGG has specific types of pili known as Spasbiae and Spicevie pili.
Spasbiae, what does that mean?
It refers to the three main protein components that make up that pili structure. There's Spaphi, which forms the main backbone or shaft of the pilus, determining its length. There's Spab, which acts as the basal anchor, attaching the pilus structure firmly to the bacterial cell wall. And then there's Spock, located at the very tip of the pilus.
And the tip is important.
Critically important. SPAC is the actual adhesin component of this structure. It's the part that physically binds to mucus components or host cell receptors. Studies using antibodies to block SPAC or using mutant LGG bacteria that lack the SPAC protein showed that this tip protein is absolutely essential for LGG to interact with and stick to mucus. Without SPAC, adhesion drops dramatically. LGG also has another type, Spaphidae pili, which also seem to mediate adhesion to mucin.
So these tiny hairs have specific parts for structure, anchoring, and sticking. It's quite engineered. What about other probiotics like Bifidobacterium?
Bifidobacterium species, another major group of probiotics, especially important in infants, also have pili. The sources mention finding various types, including type IVB tad pili and sortase-dependent pili in species like B. bifidum, B. braves, B. longum, and B. adolescentis. And these pili have been shown to be involved in their adhesion to intestinal epithelial cells or to components of the extracellular matrix.
So pili seem like a pretty common and important tool for probiotic adhesion. Does the environment affect how many pili they make? Like, do they make more if they need to stick better?
That's a really interesting point. The sources mention that environmental stress, specifically the acid stress that LGG might encounter when passing through the stomach, could actually enhance its adhesion capabilities later on.
How?
By inducing the expression of the genes related to these pilis, particularly the SPAS and SPAF genes, which encode those key adhesin components. So it's possible that surviving the harsh acidic environment might trigger the bacteria to produce more sticky bits, potentially helping them adhere more effectively if they reach the mucus layer in the intestine, a sort of adaptive response.
That makes sense. Are there any other types of surface molecules involved in sticking, besides moonlighting proteins and PLA?
Yes, a couple of others are mentioned. Exopolysaccharides, or EPS, can play a role. These are long chains of sugar molecules that some bacteria secrete outside their cell wall, forming a kind of capsule or slimy layer around themselves. This EPS layer can be involved in adhesion, biofilm formation, and interacting with the host environment. The source references Lactobacillus plantarum and Lactobacillus reuteri in this context.
So the slime itself can be sticky.
It can mediate interactions, yes. And finally, S-layer proteins are mentioned. Some bacteria, including some lactobacilli like Lactobacillus acidophilus and CFM, have a highly organized crystalline protein layer, almost like chainmail, covering their entire surface. This is called the S-layer. While primarily protective, these S-layer proteins themselves can sometimes contain specific domains or regions that are involved in binding to host structures, like fibronectin or immunoglobulin-like domains, contributing to adhesion.
It's truly remarkable the diverse toolkit these tiny organisms possess just to try and find a foothold and hold on to the gut lining. It's clearly not a simple matter of just passively floating by. It's an active molecular effort to colonize.
It absolutely is an active engagement with the host environment. And understanding these specific molecular mechanisms which adhesins bind to which host receptors is really key to figuring out why some probiotic strains might be better colonizers than others, or why they might stick preferentially in certain parts of the gut.
Which leads us to the next level of understanding. Thanks to modern science, especially genomics, we can now look even deeper, right into the genetic code, the DNA blueprint of a specific probiotic strain to get clues about its potential capabilities.
Exactly. We can move beyond just observing behavior to understanding the underlying genetic potential. The sources provide a fantastic example of this with a study that performed a deep dive into the genome of a specific strain, Lactobacillus delbrueckii subsep indica strain TY11.
Where did this TY11 strain come from?
It was isolated from a traditional native yogurt made in Bangladesh. So, a naturally occurring bacterium from a fermented food. Scientists sequenced its entire genome, read all 1,916,674 base pairs of its DNA to see what probiotic relevant features were encoded there.
What did its genetic code reveal about its potential as a useful probiotic? Did it look promising?
It certainly did. The analysis revealed a whole suite of genes that suggest TY11 is well equipped to adapt to the diverse and often stressful conditions found in the gut environment.
Okay, like what specifically? What's the first thing you look for?
Well, a crucial first check is always safety. The genomic analysis confirmed that TY11 did not contain any known genes associated with antibiotic resistance, nor did it seem to have pathways for producing harmful toxic metabolites. That's a really important baseline safety assessment for any potential probiotic. You don't want to introduce something harmful.
Absolutely. Safety first. What about its ability to handle the stresses we talked about? Acid, bile, temperature changes.
Its genome showed it has the genes to cope. It possesses genes for molecular chaperones like DNAC, GRO-EL, and GRO-WASE. These proteins are like cellular repair crews. They help other proteins fold correctly or refold if they get damaged by stress, which is vital for surviving conditions like heat shock or acid stress, and even under normal conditions.
So it has its own maintenance team.
Exactly. It also had genes for stress-related proteases like CLPX, CLPP, HSLV enzymes that can specifically chew up and remove abnormal or damaged proteins, preventing them from causing problems in the cell. And it had genes for an osmoprotectant transport system.
Osmoprotectant, what does that do?
That helps the cell adapt to unfavorable osmotic conditions like high salt concentrations or even temperature stress. It allows the cell to take up compatible small molecules from the environment, which increases the internal osmotic pressure and helps stabilize proteins and membranes. So multiple genetic systems pointing towards resilience.
Okay, so it looks genetically tough. What about how it interacts with food components in the gut? Does its genome tell us about its metabolic abilities, and could those benefit the host?
Yes, the genome provides a really detailed picture of its metabolic potential. It showed that TY11 has the complete set of genes for glycolysis, the main pathway for breaking down glucose to get energy. It also has the pentose phosphate pathway.
So it can process sugars well.
Very well, it seems. It contains genes for a phosphenyl pyruvate-dependent phosphotransferase system, or PTS. This is a specialized system many bacteria use to actively transport various sugars into the cell while simultaneously phosphorylating them, adding a phosphate group. The analysis showed TY11's PTS could likely handle a wide range of sugars: beta-glucosides, silabiose, fructose, glucidol, galactose, lactose, mannose, sorbitol, and glucose. This suggests it's quite versatile and can utilize multiple different carbon sources it might encounter in the gut.
That sounds useful. What about lactose specifically? Lots of people have trouble with lactose.
That was a key finding. The genome showed TY11 has the genes for components of the lactose PTS and also for an enzyme called 6-phosphobeta-galactosidase, laxogener. This enzyme breaks down the phosphorylated lactose that the PTS brings in.
Mm-hmm.
Interestingly, the gene for lactose permes, another way to get lactose in, seemed to be missing. But having the PTS system suggests it can ferment lactose, similar to its relative Lactobacillus delbrueckii subsep P. bulgaricus, which is used in yogurt making.
And that's relevant because
Because the ability to break down lactose in the gut could potentially help alleviate symptoms of lactose intolerance, which, as you mentioned, is a major issue for a large percentage of the global population. So this genetic capability is a significant potential probiotic benefit.
makes sense. Did the genome show anything about handling proteins or fats?
Yes. For proteins, it encoded genes for proteolytic enzymes to break down proteins and various peptide transporters to absorb the resulting small amino acids and peptides. This capability could potentially help with protein digestion and might even affect the host's energy balance through the metabolism of these protein components. It also had genes for enzymes like multicopper oxidase and nitroreductase that might help decrease the production of potentially toxic metabolites that can arise from protein breakdown in the gut.
Then fats.
For lipids, the genome showed genes for esterases and lipases, enzymes that break down fats, and also for transmembrane lipid transporter proteins called flipases. The source suggests these might potentially play a role in modulating human adiposity or body fat levels, though the mechanisms would be complex.
So its genetic blueprint really indicates it's not just a passive passenger, but potentially an active participant in processing various components of our diet in the gut. Based on all these genomic features, did the study predict specific health benefits or roles for TY11?
Yes. Based on this comprehensive genomic profile, the researchers predicted several potential beneficial roles. They suggested it could help prevent the overgrowth of harmful bacteria and aid in treating indigestion caused by them, likely by altering the gut bacteria makeup through competing with pathogens, pathogen exclusion, and successfully colonizing.
Okay.
It was also predicted to potentially help alleviate antibiotic-related diarrhea. The idea here is that by adhering to the gut lining and possibly producing antimicrobials, it could prevent harmful bacteria from taking over after antibiotics wipe out many beneficial ones and maybe help replenish the good guys.
That's a common hope for probiotics.
It is. And they also suggested a potential role in helping with complex conditions like irritable bowel syndrome, IBS. IBS involves things like altered gut flora, inflammation, and immune dysfunction, and the features of TY11 suggest it might positively influence some of these factors, although the exact mechanisms in IBS are still being worked out.
You mentioned it might fight off bad bacteria. Did its genome show specific weapons it might use?
It did. The genome analysis revealed that TY11 has the genetic instructions needed to synthesize two specific bacteriocins, one called helveticin J and another called aterolacin A.
Ah, those antimicrobial proteins we talked about earlier in the context of colonization resistance.
Exactly. By producing these bacteriocins, TY11 could potentially inhibit the growth of closely related bacterial species or other competitors vying for the same niche in the gut. This would give it a competitive advantage, aiding its own colonization efforts and helping to exclude potential pathogens.
A built-in defense and offense system. And going back to adhesion, which we said was so important, did the genome analysis find genes specifically related to sticking?
Yes, it pointed towards adhesion capabilities too. The source notes the presence of genes encoding putative, meaning likely but not yet experimentally proven in this strain, extracellular proteins. It also identified genes for components of the main protein secretion system in bacteria, the second-gig translocase and YIDC. This machinery is needed to get proteins out of the cell or inserted into the membrane or cell wall, including proteins that might be involved in interacting with the extracellular environment and mediating adhesion. So the genetic potential for sticking seems to be there.
It's fascinating how much information about potential function you can pull just from reading the DNA sequence. Did they compare 2Y11's genome to its close relatives?
They did. They compared it specifically to Lactobacillus delbrueckii subsep P. bulgaricus, the well-known yogurt starter. They found some interesting differences. There were 37 genes present in the core genome of subsp. indicus, like TY11. They were generally absent in subsp. bulgaricus. Conversely, There were 10 genes common in Bulgaricus that were either different or deficient in TY11. These genetic differences likely translate into distinct functional capabilities between these closely related subspecies. So this genomic deep dive really provides a molecular rationale, a strong foundation, for why a specific strain like TY11 might have probiotic potential. Its resilience, its metabolic skills, its ability to produce antimicrobials, and its likely capacity for adhesion are all, in a sense, written in its DNA. That's the key insight, exactly. The genome gives scientists a roadmap of the potential capabilities and limitations of a strain, often before doing extensive, time-consuming lab or clinical testing. It showed that TY11 appears genetically equipped to navigate the challenging gut environment and potentially perform beneficial functions for the host.
Okay. The gut is clearly a tough place for these beneficial bacteria to arrive and thrive. So even with all their natural adaptations encoded in their genes, like we saw with TY11, is there anything we can do from the outside using technology to give these probiotics a better shot at surviving the journey and reaching the colon in sufficient numbers?
Yes, absolutely. Since the natural survival rate can be quite low for many strains, researchers have been working on strategies to protect them during their transit through the gastrointestinal tract. The main approach discussed in the sources is microencapsulation.
Microencapsulation. That sounds like putting them in tiny capsules.
That's essentially it. It's a technology used to coat the probiotic bacteria or embed them within a protective matrix material. This creates a physical barrier, a tiny shell or bead, around the cells.
And the purpose of this shell?
The primary purpose is to shield the probiotics from the harsh conditions they encounter, particularly the strong gastric acid in the stomach and the damaging bile salts and digestive enzymes in the small intestine. The idea is that the capsule protects them through these hostile zones.
And then what happens? Does it open up later?
Ideally, yes. The capsule material is often designed to remain intact in the acidic stomach, but then dissolve or degrade under the more neutral pH conditions found later in the intestines, or perhaps be broken down by enzymes present there. This releases the viable bacteria closer to their target site in the colon, having protected them through the most dangerous parts of the journey.
So it's like an armored transport vehicle for bacteria. What kind of materials and methods do they use to create these tiny shields?
The sources mention several different approaches and materials, often using natural polymers that are safe for consumption. We already touched on some gels used in SEM prep, like agar gel for viscous foods or calcium alginate gel for heat-sensitive ones. These can encapsulate bacteria. For more dedicated microencapsulation, alginate derived from seaweed is very commonly used. You typically mix the bacteria with the sodium alginate solution, and then extrude this mixture as droplets into a solution containing calcium ions. The calcium causes the alginate to cross-link and instantly form gel beads or capsules around the bacteria.
Okay, alginate beads. What else?
Gelatin is another option, sometimes combined with other chemicals like toluene diisocyanate to form stronger, more crack-tolerant capsules. The source mentions Bifidobacterium pseudolangum, freeze-dried and coated this way, then with wax showed higher survival in simulated gastric juice. Chitosan, a polysaccharide derived from shellfish shells, is also used. It's soluble at low pH but can form gels, often used in emulsion methods to create beads.
Emulsion methods?
Yes, that's another major technique. You basically create an emulsion like oil droplets dispersed in water or water droplets in oil, with the bacteria and the coated material in one of the phases. Then you solidify the coated material within the droplets to form microcapsules. This often involves filtering to recover the microbeads afterwards. With alginate emulsions, they might use a fat-soluble acid to trigger gelation. Using emulsifiers can help create very small beads down to 25-35 micrometers.
Are there industrial scale methods?
Spray drying is mentioned as a common industrial scale method. Here, you mix the bacteria, often fresh, not freeze-dried, into a liquid protective matrix, maybe something like heat-treated whey protein with milk fat droplets, and then you spray this mixture into a stream of hot air. The liquid evaporates rapidly, leaving behind dry, encapsulated particles containing the bacteria.
Sounds efficient, but maybe harsh.
It can be. Controlling the temperature and process is critical to minimize heat injury to the cells during spray drying. Similarly, if you use high-energy processes like jet mills for making emulsions, the excessive shear forces can damage the bacteria. So process optimization is key for all these methods. There's also a chopping system method mentioned, using a matrix of fat, flour, and starch to form larger shelf-stable microcapsules, 0.5, 1.5 millimeter.
So lots of different ways to build these protective homes for the bacteria. Are there any downsides or challenges with using microencapsulation?
Yes, there are practical considerations. For example, if you're using encapsulated bacteria as a starter culture to make fermented foods, like yogurt, the fermentation process might take significantly longer. Because the bacteria are initially trapped inside the capsules, they can't start multiplying and producing acid freely in the milk right away. They have to be released first, which slows things down compared to using free cells.
That makes sense. Anything else?
Cost is another factor. Adding a microencapsulation step obviously adds complexity and expense to the production process compared to just adding free probiotic cells to a product. And as mentioned, you have to be careful that the encapsulation process itself doesn't harm the bacteria you're trying to protect. Finding the right balance between protection and viability is crucial.
And how do scientists actually know if the encapsulation is working effectively? How do they measure how many bacteria survive the process and the simulated gut conditions?
Assessing viability is absolutely critical. They typically do this by taking the encapsulated product, exposing it to conditions that mimic the stomach, low pH acid, and then the small intestine, neutral pH with bile salts and enzymes, for relevant periods of time.
And then they count the survivors.
Exactly. After the simulated digestion, they'll try to release the bacteria from the capsules, if they haven't already dissolved, and then plate them onto specific culture media in the lab to count how many living cells are left, usually reported as colony-forming units, or CFU, per gram or milliliter.
Is there a target number they aim for?
The sources mention the concept of a dielectric index, DI, as a measure, and note that a commonly recommended minimum dose for probiotic efficacy is often around 1 billion viable cells per day reaching the gut. So encapsulation aims to ensure a much higher percentage of the initial dose survive to contribute to that target number.
And counting them is straightforward.
Well, there can be nuances. The sources mention that the type of culture medium used to grow and count the bacteria after stress or storage can influence the recovery rate. Some media might be better at helping slightly injured cells recover and form colonies than others. Also, they point out that the loss of viability, say, during storage of a fermented product like yogurt, isn't always a simple straight line downwards. The conditions inside the product, like pH, accumulating acids, are constantly changing as the bacteria metabolize, which affects their survival rate over time in complex ways.
So microencapsulation seems like a really powerful technological strategy to help probiotics overcome the significant biological challenges of the GI tract, but it's clearly a complex field with its own set of technical hurdles and optimization challenges.
It definitely is. It's a major focus of research and development in the probiotics industry, finding better, more effective, and more cost-efficient ways to protect these beneficial microbes and ensure they get delivered alive and kicking to where they can potentially do their job.
Well, this has been quite a journey today, a real deep dive into the often unseen world of bacteria. We started by looking at the intricate and sometimes surprising techniques scientists have to use just to visualize these tiny organisms, navigating challenges like potential artifacts and handling vastly different sample types using methods like SEM.
We certainly did. And then we followed the incredibly demanding journey that beneficial probiotics face when consumed, highlighting those formidable barriers, the harsh stomach acid, the digestive enzymes and bile salts in the small intestine, and then that really intense competition from the established gut microbiota, that phenomenon of colonization resistance in the colon.
Yeah, that resistance is key. We explored the clever molecular strategies these bacteria use to try and overcome those challenges, focusing on how they actively seek to adhere to the intestinal lining. We discovered this diverse arsenal of adhesins they employ, from those unexpected moonlighting proteins with hidden sticking talents to specialized structures like pili acting as grappling hooks, and even contributions from surface layers like EPS and S layers.
And we saw how powerful genomics can be, delving into the molecular potential of that specific strain, TY11. The genomic analysis provided this fascinating blueprint, revealing the genetic basis for its likely stress tolerance, its versatile metabolic capabilities, its ability to produce antimicrobial bacteriocins, and its potential for adhesion, all giving molecular insights into its suitability as a probiotic.
Finally, we looked at how technology, specifically microencapsulation, is being developed and refined as a strategy to give these beneficial bacteria a crucial helping hand, shielding them from that harsh GI environment to significantly improve their chances of survival and successful delivery to the gut.
It's abundantly clear, isn't it, that understanding probiotics is an incredibly complex endeavor. It requires knowledge spanning microscopy, bacterial physiology, gut ecology, microbial genetics, host microbe interactions, and even material science and engineering for the delivery technologies.
Absolutely. It's this intricate interplay between the inherent traits of the bacteria themselves, the highly dynamic and challenging environment of the host gut, and the technological interventions we design to try and bridge that gap.
And the research is definitely ongoing. Scientists are constantly working to better understand the precise mechanisms of adhesion for different probiotic species, figuring out exactly how they interact with that incredibly complex resident gut community to overcome colonization resistance and developing smarter ways to ensure their effective delivery.
This deep dive really pulls back the curtain, I think, on the sophisticated science that underpins the whole concept of using beneficial bacteria for health. It's far from a simple idea. It's really about understanding survival strategies, intricate biological interactions, and clever engineering solutions, all happening at a microscopic scale.
It truly underscores the complexity and the elegance of these microbial systems and our attempts to work with them.
Absolutely. It makes you think about the incredible biological engineering happening constantly within us and all around us. And considering that point you raised earlier about how colonization efficacy seems to vary so much between different people, some individuals being more permissive to probiotics, while others are quite resistant.
Yes, that individual variability is a really fascinating aspect. It really brings up some interesting possibilities for the future, doesn't it?
It really does. Could we potentially reach a point where, instead of just recommending general probiotic strains off the shelf, scientists could actually analyze your specific gut environment, perhaps your unique microbial community structure, maybe even aspects of your genetics or gut physiology, to predict which specific probiotic strains are most likely to survive, adhere, and potentially thrive in your particular system? What might that level of personalized microbial intervention look like? How could tailoring probiotic recommendations to the individual change the way we think about using bacteria for health in the future?